So good morning, everyone. So welcome to day 3 of our special lecture series. So we are thrilled to see such a diverse group gathered here today. So it's a privilege to host you for what we anticipate will be both enlightening and engaging. So today we are diving into the fascinating world of therapeutic protein production and purification in the biotech industry. Our esteemed speaker is ready to share their deep knowledge and valuable insights on this complex topic. So we encourage to dive into the discussions, ask questions, and fully embrace this learning opportunity. So thank you for joining us. We are excited for these stimulating conversations and insightful takeaways that lie ahead. Let's make this a memorable and impactful experience together. Now, I would like to call upon Dr. G. Saravanan, Dean, School of Sciences and Humanities, to deliver the welcome address. So, welcome you, sir.
Good morning.
So dear participants, sorry for the technical issue. Please be wait. So now I would like to call upon Dr. G. Saravanan, Dean, School of Sciences and Humanities, to deliver the welcome address. So over to you, sir.
Esteemed guests, participants, and colleagues,
Sir, excuse me, sir. Sorry for the technical issue. So there is a technical issue from our organizer side. So there is a deep apology for that.
true honor to welcome you to
So dear participants, if there is a technical issue, please be patient. We'll be back. So sorry, sir. Dear participants, apology for the technical issues. So now I would like to call upon Dr. S. Vadivakarasi, Assistant Professor, Department of Biochemistry, to deliver the welcome address. Welcome you, ma'am.
Sir, good morning, sir.
good morning, madam.
So good morning to all. Good morning, esteemed guests, participants, and colleagues. It is a true honor to welcome you to day 3 of our special lecture series. I am delighted to have you join us for what promises to be an exceptional and insightful day. Today, we are privileged to have Dr. J. Venkateshwara Rao, a renowned professor in zoology from Osmania University, as our resource person. Dr. Venkateshwara Rao brings a wealth of knowledge and expertise, particularly in the field of therapeutic, protein production, and purification, which is the focus of our discussion today. Dr. Venkateshwara Rao extensive research and academic contributions have significantly advanced our understanding of biotechnological processes. We are fortunate to have him share his insights and experience with us. His expertise will undoubtedly provide us with a deeper understanding of the complexities and innovations within the biotech industry. I encourage you all to take full advantage of this opportunity, engage with the content, ask questions, and participate actively in the discussions. This is a valuable chance to learn from a leading expert and to expand your knowledge and perspectives. Thank you for being here, and let's look forward to a day of inspiring discussions and valuable learning experience. Welcome, and let's make this day a memorable and enlightening one. Sir, welcome you, sir.
Thank you, madam.
Thank you, ma'am. Thank you so much. So now it's time to introduce our resource persons. So thank you, sir. First of all, I would like to thank you for accepting us our invitation. Uh, Good morning, everyone. So it is distinct pleasure to introduce our esteemed guest and resource person for today's session, Dr. J. Venkateshwara Rao. So he is an eminent professor in the Department of Zoology at Osmania University, Hyderabad, with a remarkable academic and research background. So Dr. Venkateshwara Rao is a distinguished academic and researcher currently serving as a professor in the Department of Zoology at Osmania University, Hyderabad. His extensive educational background includes MSc and B.Ed, Bachelor of Educational degree, a Master of Technology, M.Tech in Biotechnology, and Doctor of Philosophy from Jawaharlal Nehru Technological University, Hyderabad. He has also earned advanced diplomas in bioinformatics, patent laws, and computers, reflecting his diverse expertise. So, Dr. Venkateshwara Rao's academic journey began with notable achievements in his early education, including a high distinction in SSC and intermediate studies, and first class honors in this undergraduate and postgraduate degrees. His scholarly pursuit continued with a prestigious Boys Caste Fellowship from the Department of Science and Technology, Government of India, and postdoctoral research at Virginia Commonwealth University, USA. Professionally, Dr. Venkateshwara Rao's career encompasses a wide range of roles and responsibilities. He began with an internship in plant genomics at ICRISAT, Hyderabad, and has since held significant positions such as head of the biotech department at Mallareddy Engineering College, Hyderabad, assistant professor at JNTU, Hyderabad, and assistant professor at Osmania University. His expertise extends to visiting professorships and academic coordination roles at various institutions, including IICT, MHRD, and Nagarjuna Government Degree College. His administrative experience includes serving as a vice principal and control of examinations at Nizam College and academic coordinator and nodal officer for All India Survey on Higher Education (ISHE) under MHRD, Government of India. So Dr. Venkateshwara Rao's research contributions are substantial with involvement in several high impact projects funded by UGC, DBT, AP cost and received fund of more than rupees 1 crore 70 lakhs. His research areas including biotechnology, gene therapy, bioinformatics, and environmental sciences. So, notable projects under his leadership include studies on recombinant proteins, gene therapy for tumor angiogenesis, and bioremediation of fluoride compounds. He has authored several influential books and numerous research articles published in leading journals. His publications cover a broad spectrum of topics from molecular biology and bioinformatics to environmental and medical sciences. So Dr. Venkateshwara Rao has also been actively involved in organizing and participating in various workshops and conferences, contributing to the scientific community's understanding of emerging trends and technologies. So Dr. Venkateshwara Rao's exceptional contributions to science and education have been recognized through numerous awards and honors, including the topper in pre-PhD examination JNTU Hyderabad and UGC CSIR NET Award and prestigious Boys Caste Fellow Award and the International Research Excellence Award by the Global Indo-US Summit. His commitment to advancing scientific knowledge and education is evident in his extensive involvement in academic activities, including serving on various board of studies, examination committees, and research project evaluations. Dr. Venkateshwara Rao is not only a respected educator and researcher, but also a dedicated contributor to the advancement of science and technology in India. His work continues to inspire and influence the fields of zoology, biotechnology, and bioinformatics. So we are fortunate to have him today session. So we are welcome you, sir. We are happy to be here. We would like to thank you for accepting our invitation to take part in this online lecture series. Our participants and our organizers are fortunate to have you here, sir. So the session is now yours. Welcome you, sir.
Thank you, sir. Thank you, Dr. Sati Babu Uddan Rao, for warm welcome for me for your institution, K.S.Rangasamy College of Arts and Science, for being a part of your time with me. So 1.5 hour or 1 hour, whatever the time I want to spend with young minds, young budding scientists, so that I can help them in clearing their doubts or their future perspectives. So whatever they want to plan, they can have a plan, they have a clear thought. So the transparency is going to maintain throughout the lecture. So thank you very much, sir, for the whole management of K.S.Rangasamy for inviting me to develop my lecture, my sharing my expertise with all the young minds here. And also, it is no, as human beings, we can do any wonders, especially there is huge area is there, like in life sciences, therapeutic proteins. I'm going to select only one area today, lectures, because you are all turning the 4 days lecture series. One of the important and application, job-oriented application is also a point where we are going to touch the industry is therapeutical applications. So I want to enlighten in that aspect. So before going to that, I am seeing the participants number is about 85 is there on screen. So almost it is going to reach 100 also. So can I see your cameras? Can is available at least a few of you faces I want to see, student faces. Is it possible? Two students, if says good morning or says that your name, that is enough. Showing your face and uh, saying your name, that's it.
Morning.
Okay, no problem. If one or two says okay, shows your face, that's fine. Okay, you can unmute one of you. And what is your ambition in your life? Why you have chosen life sciences? Why you have chosen biochemistry, or why you have chosen biotechnology like that? You can express one or two words. You want to become scientist or professor, or what you want to become?
So, sir, achievements, games, the portal. Up to date.
Participants, please mute your mics. So, hello, sir.
Yeah, please. Or is it audible for all of you? Is it audible?
Yes, sir. You are audible, sir.
Okay, okay.
So
Is
please
anyone
share
is
your
going to answer what is your life ambition, what you want to become in future? So you're studying MSc Biochemistry or MSc Botany Zoology,
Good morning.
Chemistry. Yeah, please, one of you share your.
So I'm USG from Satyabama College, Chennai. I'm
Very
studying
nice.
UG Bioinformatics with Data Science, sir. My future
Very nice.
ambition is I want to be a researcher in influencing AI in biology, sir.
Very good option. Very nice. I will appreciate you. Is any one more? So why you are studying this particular courses like biotechnology, bioinformatics, no, biochemistry, microbiology, genetics? Why you are studying? What is your aim? Any other person? Feel free to express what you want to become. Is anyone is there to become scientist here or professor?
dear participant. Please feel free to interact with the resource person. So he's such a nice interactive person.
Please raise your hands. If you want to become professor, you can raise your hand. And the right corner, you can have raise is there. You can click the raise. How many of you want to become professor? How many want to become scientist? The reason why I am asking this question is that if you love your profession, then only it's possible to make the steps to reach your goal. If you don't love your profession, don't express your thoughts, vision, it's very, very difficult to give training also. So forget about those things, but give a feedback to your teacher after this session is over, what you want to become. If you want to become professor, there is a lineup. If you want to become industrialist, you want to pursue jobs in industry, there is another way. If you want to become pure researcher or a scientist, there is other way. So anyway, you are pursuing science, you are loving science, you have a lot of interest in science. Let us make this session very interactive with you. So how many have heard the word cell? It may be animal cell, bacterial cell, plant cell. How many of you heard the name cell and observed under microscope? Second, how many of you have seen the using the microscope, the nucleus? How many of you seen nucleus? How many of you seen the chromosomes? How many of you seen the genes? How many of you seen the DNA molecule? Then coming to main question, how many of you are interpreting the information in the DNA molecule? So I want to ask one question. What is the composition of DNA? Please, any one of you. What is the composition of DNA molecule? It is adenosine, thymine, guanine, cytosine. With only 4 letters, the DNA molecule is made up of. So this is the information which is there in every living organism. Is it correct? DNA is made up of only 4 nucleotides, ATGC. Is the information is correct or it is wrong? No. Okay, let me come to a conclusion. Yes, Gita has told it is yes. So ATGC, with ATGC letters, there are so many life forms in this universe, and there may be plants, animals, ranging from small bacteria to largest animals on this planet share this same common information, common genetic code in the form of ATGC. So the proteins are also, which are derived from the DNA, is having certain type of amino acids. The combinations of amino acid makes the peptides or proteins. So our body is made up of all the proteins, enzymes, hormones, and related molecules, macromolecules. So DNA, RNA, and proteins, enzymes, hormones, all these small peptides, molecules are going to make your tissues, your organs. The complete living organism is a biochemical machine. So this biochemical machine is going to work on certain principles. It is going to work on genetic principles. It works on certain biochemical reactions. Because of these biochemical reactions, you are in the living state. So what is the difference between a living and non-living organism? Some biochemical reactions are happening in your body from time to time. It is taken care by inbuilt mechanism, which is there in the form of DNA molecule. The DNA is a machinery, which is a blueprint of your existing machinery, and the commands, the instructions which are passing from the DNA is going to reach to the ribosomes. Ribosomes are going to prepare the protein machinery. The protein machinery, which is synthesized or manufactured in your body, is going to work this mission as perfect mission. So all organisms are going to work with the help of the DNA machinery. DNA machinery is going to produce a lot of enzymes, hormones, and peptides. So right now, because of the invention of the DNA molecule by 1953 by Watson and Crick, now we realize that the DNA double helical model, how it is there inside our cell, inside the nucleus, how it is having the genes. So how many genes are there in human being? Anyone is having any idea? How many total number of genes are there in the human body? How many genes approximately? A data scientist is there, Geeta. How many genes are there? Approximately in our body, there are 32,000 genes are there. Among them, 3,000 genes are very functional. 3,000 genes functional means that every time they are going to produce the enzymes, hormones, and all types of regulatory mechanisms they are going to take care of. So some of the genes are known as housekeeping genes. These housekeeping genes are going to make you alive. For example, you are breathing uncontrollably, you are taking your breath. Nobody is controlling your breath, but there is a mechanism which is inbuilt in your genes, which is there. With the help of these genes, the your brain part is active. As long as your medulla oblongate is active, you are taking the respiration, you are breathing. So this part is already inbuilt mechanism is there in your genes. It is going to take care of. Like the so many mechanisms are there in your body, which is going to take care of respiration, digestion, excretion, respiration, reproduction. So many systems are there. Nervous system. Every system has some kind of function. It is going to take care of everything in your body. So yeah. So I'm going to share my PPT over here. Please look into that particular PPT. Can you able to see this PowerPoint on your screen?
Yes, sir. It is visible, sir.
Okay. It is today's topic is therapeutic proteins. So I'm going to highlight some of the concepts over here.
Sir,
But before
excuse
going
me,
to
sir.
this concept, yeah, before
Sir,
going to this
excuse me.
concept, I want to show you one paper, one important paper. You can note down, if you have paper and pen, you can note down this particular paper, Evolving Paradigms of Recombinant Protein Production in Pharmaceutical Industry, a rigorous review. In this, I'm going to bottom page of the paper. I'm going to show you, highlight some of the things. Some of the products of the therapeutic proteins. I'm going to highlight those things. This table is it visible? Recombinant biopharmaceutical manufactured with microbial host. Is the table is visible?
Yes, sir. It is visible, sir.
Yeah. See that some of the products. Some of the products I can highlight. For example, insulin. its trade name is Himalayan, cell fact is E. coli, application is diabetes, company name is so and so. Okay, like that you can see some of the products here. Some of the products you can I want to highlight. You can see next is growth hormone. You can see its trade name, application, pituitary dwarfism, so and so company. Like interferon, hepatitis B vaccine.
நான் ஃபாலோ பண்ணியா
Please mute yourself.
கவின்
See,
Dear
next product is, you
participant,
can see,
kindly mute your mic. Dear participant, kindly mute your mic.
what you have to focus here is that what is the product over here. There is some problem in the application. Even then we'll try. What you have to see is that the product name and the company name and what is its application. You can see here, just you can read with your eyes itself. First product is insulin, second product is growth hormone, third product is interferon, fourth product is interferon beta 1b. So many products are there, just I am highlighting only few of the products. Next is granulocyte colony stimulating factor. On this product I worked for my PhD work. Next is granulocyte macrophage colony stimulating factor, interferon gamma protein, pegylated interferon alpha 2a. Like that, there are hundreds of proteins are there, protein products are there, and the corresponding company names are there. Like that, you can have this list in the paper. There are several hundreds of proteins, like for example, somatotropin, semaglutide, vorocetide. Like that, thousands of compound molecules are there, which are the products of the biotechnology or bioinformatics, or you can say biotherapeutics. So these compounds, like if you want, I will send this file to you. Of course, some problem is there in the PDF. It is opening and closing. So like that, for example, you can see here, human serum albumin. Trade name is Medway. Cell fact is Pastilla pastoris. Produced by so you can see here some of the compounds. You can take any compound, of course, don't worry. For example, you are taking here some compound vorocetide, Voxzoro, E. coli, like that. So this is an example. What I'm saying is an example. Like that, you can have thousands of proteins which we can produce by using the normal way of molecular biology, microbiology, and using the biochemical methods. So after seeing this, after seeing these products, what is your idea? Just tell me, any one of you can say some words, few words about these products and what is the idea behind these particular products. For example, any one protein you can take. For example, first protein is Sargastomostism leucan Saccharomyces cerevisiae. After seeing all these products, what is your inference? What is your conclusion? Tell me in one word. Are you able to see these products on your screen? It is going on, of course, don't worry about that. But these are the thousands of products which are coming into the pipeline by biotechnology industries. What is the difference between allopathy to biotechnology industry? Do you know some idea about allopathy or medicine? Have you heard the word allopathy? How many of you heard the word allopathy? So you should have a basic knowledge when going to for research work. Allopathy is now what you can call as English medicines. All English medicines will come with some kind of side effects. So now the world is moving to the world where the side effects are very negligible or zero. So here therapeutic proteins are the natural products which are produced from your body and you are going to treat the diseases. So here, for example, insulin is very short in the diabetic patient. You are going to produce insulin on artificial way by using this technology. So like that, so many products like interferons, interleukins, monoclonal antibodies, all types of therapeutic proteins, all this kind of stuff we can produce by using the biotechnology industry. So this is a replacement of the allopathy, where there's a lot of side effects are there, where compounds are unknown organic compounds with lot of side effects that may cause cancers also in some cases. So these therapeutic proteins are the natural ones which are derived from your own body cells, which are the products of your own cells. So these proteins, when engaged in a proper way, they are going to cure some of the diseases or genetic disorders or some kind of metabolic errors. So because of the seeing the importance, now the pharmaceutical industry shifted from the chemical industry to pharmaceutical sector to the biotechnology sector, where there is a lot of
Sir,
investment is there.
excuse me, sir.
Yeah,
Sir, excuse me. Sir, please put your presentation on presentation mode, sir. So
yeah,
slides
sure,
are
sure.
not moved.
Is visible, sir, this one?
So, you put it in presentation mode, sir.
Yeah, I
Yes, sir.
kept it
Yes.
presentation.
Yes, sir.
Is it
Yes, sir.
visible?
Now
Yeah,
fine. Now fine, sir.
right.
Yes, sir. Thank you, sir.
So we are going to go with introduction, because once you know the importance, then only you can realize the importance of these products. First is, let us see the introduction part. Proteins which are engineered in the laboratory for pharmaceutical use are referred as therapeutic proteins. Please note down this word. The proteins which are engineered, that means using the protein engineering methods, what you want to modify, way to make them active, which active recipients are active in controlling the disease or disorder, that proteins you are going to synthesize in laboratory for pharmaceutical uses, those are known as therapeutic proteins. So next slide. Proteins which are absent or low in individuals with an illness such as cancer, infectious diseases, hemophilia, anemia, multiple sclerosis, hepatitis B and C, or artificially synthesized on a large scale through genetically modified cells and are going to be delivered. So this therapeutic approach is in treating diseases using proteins and peptides is termed as protein therapeutics. Protein therapy is similar to gene therapy, but unlike gene therapy, protein therapy delivers proteins to the body in specific amounts as would ordinarily present to help repair illness, treat pain, or remake the structures. So as we already know that our body is going to function with a lot of enzymes, hormones, and proteins. So here the gene therapy or protein therapy is very, very similar to gene therapy, but exactly the dosage we are going to mention in terms of protein products, and you are going to treat the disease. So example, you can say it is in the protein therapy, like in the form of, it is introduced in 1920. Human insulin is considered as first therapeutic protein. So some of the diabetic patients, they will be treated as artificial human insulin from 1920 onwards. So like that. So these peptide therapies are more advanced than gene therapies, and they are now they are people are using these therapies to replace the old traditional classical what you can say allopathic treatments. So proteins are considered by the following facts: diversity of the functional groups. There are so many diverse groups are there in the proteins, like free thiols, which are having cysteine residues, amine residues. You can able to convert these residues by using the protein engineering methods, and the structures are very, very simple structures, and they are having very few residues. So the components are not much complicated like chemical compounds. They are very, very straightforward. So next point is they're having very low side effects when compared to allopathic medical reactions. You don't know if you take the treatment for fever or headache, you are going to have the effect on the liver. If if liver is not affected, it may affect on the kidney. If kidneys, if you want to treat kidney, then if you want to treat the kidney, it may have effect on the heart. So like that, there is no specific action of the allopathic drugs. They can act on any tissue, on any organ. So a lot of side effects are there with the English medicines. But here, the proteins are going to work on very specific targets. Because of that, there is lower side effects. So that is one advantage. And they are very, very, very similar to biological components. As they are very similar to biological components, the 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 negative reactions are very less. So next is, they are less likely the body to evoke immune responses, and that naturally produces many of the proteins. So these are easily can get approval by the FDA approvals completely in a very faster way than allopathic drugs. So these are the advantages, some of the advantages where the proteins are going to express their therapeutic. For example, as a common fact, before going to that, For example, you have here, yeah, how many of you know that about hemoglobin? What is its function? Tell me in one word. Hemoglobin. What is its function? Any one of you please say, what is the function of hemoglobin? It is meant for oxygen transportation. It is going to deliver the oxygen to each and every cell in your body. So that is where the hemoglobin is there in RBC, and it is going to catch the oxygen and is going to transport oxygen to each and every tissue or cell inside your body, and it will pick up the carbon dioxide and you are going to release to outside. So the simple action of the protein, the hemoglobin, which is there in your body, is going to support for your life, for your respiration process. So if the hemoglobin is not there in your body, what is the fate of the body? The body will die. So if hemoglobin is not functioning in your body in a very proper way, the body is going to die. So here there are so many proteins which are involved in the cardiovascular diseases, the proteins which are involved in the hearts, which are involved in the walls of the heart, like that. So many proteins are, a combination of the proteins are going to work for the heart function, liver function, kidney function, brain function, nervous system function, and reproductive functions. So the combination of the proteins are going to make the functions. So if any one of the protein is impaired, that has a lot of effect on the particular system. So every protein has its own specific importance. So as one of you told that in bioinformatics, you are going to see the structure of the proteins in detail, the way the critical ingredients are there. Those ingredients are very, very major functional part of the protein, and that's going to support for the its functionality. How many of you seen oxygen with your eye? Tell me. How many of you seen oxygen with your eye? Nobody, no? So then how the hemoglobin is seeing oxygen? So your hemoglobin knows where oxygen is there. In atmosphere, it is 21%. So the 21% oxygen is going to be picked up by your hemoglobin molecule. In the hemoglobin, one particular element is there, metal element is there. That metal ion is iron ion. So Fe plus 2, Fe plus 3 is able to convert. It carries the oxidation reduction reactions. When it is FeO2, FeO condition to FeO2 condition, it is going to deliver oxygen to your tissues. So during the oxidation process, it will pick up the oxygen. During the reduction process, it's going to deliver the oxygen to your tissues. Vice versa, the carbon dioxide and carbon monoxide is picked up by your Fe atom, iron atom, and is going to transport these gases across the tissues. So one simple molecule, to manufacture one simple molecule, the nature itself is taken some billions of years of evolution. Simply from non-oxygenated pigments to oxygenated pigments, like aerial animals, like what we are breathing with air, it takes millions and billions of years of evolution. Even inside the computer, by in silico approaches to devise a molecule like hemoglobin, it takes minimum billions of years of time. You can imagine the complicated structures which are involved in the evolution of the simple the proteins. A protein which is there in your body, for example, collagen is there, myosin is there, actin is there. So many proteins are there in your body. Each protein has its own function, and you cannot replace this function of the proteins just by simple other molecules. For example, your eyes are having very beautiful vision because of the rods and cones. Rods are going to give vision during the night, and cones are going to give the vision for color perception. So every element in your body is very, very sophisticated, and the sophistication came through the fine combination of the your proteins. So like therapeutic proteins also, your body is going to protect your proteins, enzymes, hormones are going to protect your body because they are evolved in a very, very specialized way. For example, here cardiovascular disease, you can address cardiovascular diseases because you know the composition of each and every arteries, veins, how they are going to cleave the cholesterols over there, how they are going to cleave the LDLs, FDLs, HDLs, like that. So every combination you have to study, then you can able to see the therapeutic application of the protein. So some of the examples you can take classification of these proteins. For example, replacement of the protein which are deficient. Example is Ipsibira, Increlex. These are the commercial compounds. Augmentation of existing pathway. Example, Neupogen provides a novel functional activity. Myoblock and protein therapeutics in a special targeting activity which interfaces molecular organism. Avastin delivers other compounds to proteins such as radionuclei, ONTAC. And next is very important, protein vaccines. Example you can take which works against the foreign agent Engirix, treatment autoimmune diseases like Rofellac, and like that. There are protein diagnostic like JRF. So many classifications are there based on which kind of application you want to carry with this kind of proteins. For example, how many of you heard that Covaxin vaccine? A little bit I am stop sharing here because to have some practical knowledge with you. How many of you heard the Covaxin? Covaxin vaccine. How many of you heard Covaxin vaccine? To treat COVID, we have developed a vaccine that is known as Covaxin, which is mRNA-based vaccine, so which works really because of the contribution of Bharat Biotech, which is there in Hyderabad. Millions and millions of people have saved their life. This Bharat Biotech is exporting the vaccines to 75 countries, including Africa. They given millions of doses at free of cost. Because of that, its MD Krishna Ellagaru, he been awarded Padma Bhushan and Padma Vibhushan by our Prime Minister Narendra Modi. So what is the reason for these therapeutic proteins? You can apply them as vaccines also. So many vaccines like foot and mouth vaccines, hepatitis B and C vaccines, like the Covaxin vaccines. Now monkeypox is coming. So you should have current contemporary knowledge on what you are going to do for the healthcare to take care of the healthcare of the individuals. So because of the designing is a part of therapeutic protein production. So on what basis you are going to design your products, on which disease you want to treat. So that is very important. So during my PhD work, we worked together with Bharat Biotech. Some of the scholars used to come to our lab to JNTU. We used to design by genetic engineering tools, by using vector engineering. We have designed some of the tools for the hepatitis B and C vaccines 20 years back. Then you have to realize that in 2019, 20, 21, we are affected with the virus. By the time they know how to synthesize the protein molecules using virus molecule. So they synthesized the vaccine, the Covaxin vaccine, and they released it into the market. So priority of the art, prior knowledge of the compounds are very, very essential. If you have knowledge, please share the knowledge with your teacher, with your university, with your college. The products line, what I have shown long back in the document, what I have shown is thousands of compounds are there with the very good supporting system of the industries where they got patents, publications, and everything. As a student, maybe you are BSc student or MSc student, it doesn't matter. But you have to refer the compounds, how they are going to work, what is the mechanism, how they are producing on industrial scale, how to produce them, what is the technology, what is the supporting mechanisms are there. If you apply your resume, whether you can get a job or not in that particular industry, or that you have to check it out. This is not going to be done by your teacher. You have to do yourself. As a student, you have to do yourself. That's what in the initial class, in the initial topic itself, I asked you, what is your ambition? What you want to become? If you want to become professor, you have to work very hard. If you want to become scientist, you have to become the state of the art of the technology. Then only you can able to execute a simple experiment or a complicated experiment. Simply coming to the class or listening to anyone will not work you. You have to work. You have to exercise yourself. So I've shown those compounds. The compounds has a history, very big history in terms of publications, in terms of patents, in terms of how it is going to work and how they have taken the FDA approvals. If you know the things, then you can able to coordinate with some of the best minds, best scientists in the world, best scientists in the India, and you can able to publish some of the data where you can present in various conferences and publications. So your knowledge should be, you have to explore your knowledge, expand the horizons of knowledge for some of the its applications. So again, we'll go back to our topic. Yeah, we'll go back to our topic. So these are the classifications based on your, your ideology. You can able to have certain framing, and these are the methods to use to produce the recombinant proteins. There are so many platforms you can select based on your vectors and host systems. Like you can take the E. coli system, you can take the Saccharomyces cerevisiae, and you can produce even by using the mammalian cells like Chinese hamster ovary cell bioreactors. There are kidney cell lines, liver cell lines, and there are so many cell lines are there. Like that example is Chinese hamster ovaries bioreactors. Next is animal bioreactors. You can produce by using the recombinant therapeutic proteins in the milk of the transgenic animals. Example is your cows, sheep, pigs. So which platform you want to produce recombinant proteins? You can choose either the microbial platform, or you can choose the mammalian cell platform, or you can choose the animal bioreactors. So this is the rDNA technology. First, you have to select your gene of interest. Then you have to integrate into the plasmid. Through plasmid, you have to take care of restriction enzymes. You have to make restriction cuts. Then you have to stick the ends with the ligase enzyme. Then you have to select a suitable organism like bacterial cell or yeast cells, or some kind of Pichia pastris like that. Then you have to clone that. You have to see the expression, and you have to grow on the antibiotic medium. From antibiotic medium, you have to select the right combination of the recombinant. That recombinant you have to make a purified protein. The protein you have to check. So the whole process is going to apply as a common blueprint to any of the recombinant proteins. So depending upon your product, you have to select the right vector, right host, and right purification method to purify your components. So next is your microbial reactors. So this is the whole microbial story is going to start with the bacteria, yeast, how you are going to, your genes are going to compatible with these bacteria, because some of the codon uses tables you have to make use for this compatibility, because prokaryotic system is different, eukaryotic system is different. If you want to express eukaryotic proteins in prokaryotic system, you have to match the codon match. Codon uses tables you have to take help, and you have to carry the cytokine analysis. Then you have to express the proper way of the proteins. So some of the limitations, every protein cannot express in every microbial vectors, because these are prokaryotic and you are expressing eukaryotic genes. So some of the complicated proteins have the problem of the folding. So proper folding of the proteins is very important for their functionality. So refolding mechanisms you have to learn, and one of the best refolding mechanism is using the urea and thiourea methods. So those things you have to take care, and also you have to take care of the N and O linked glycosylations, Y carboxylations, and proteolytic processing. So when you are going to have bacterial platforms, the glycosylation process is a very separate process, and methylation process is a very separate process for the mammalian genes. So you have to take care of those things. Then things will be expressed. So this is long back in 1920s or 1940s, we have produced human insulin. This is the, we hijacked the DNA content of the human from the pancreatic cell. Then we have done a recombinant DNA by using a proper vector. Then we expressed in a proper E. coli bacteria. Now we are producing the human insulin by most of the companies like Roslin, like companies. So it's worldwide. Diabetic patients are using these insulin shots or insulin hormone treatments. So it's happening. This is happening with many products. Many products. So how to purify these compounds? Again, there is a separate, as your Department of Biochemistry know very well how to purify the components. You have a lot of chromatography techniques are there, lot of spectroscopy techniques are there for analysis of the pure compounds. Then you are going to see the purity, quantity, and quality and characterization studies. Then if they are pure, 100% pure, then only the FDA approvals will be given, and you are going to get patents for that, and that is going to get into the therapeutic line. So these are things. First you have to see your uh, first you have to see the, for example, in this case, the diabetes is a disease. So how to treat the diabetes? There is a hormone, insulin hormone is there. Insulin hormone is a protein. So protein therapeutic is going to come into the plane. So proteins behind the machinery is your genetic code. So DNA is going to come into the picture. From DNA, you have to isolate the gene. Then you have to produce the gene. From gene, you have to produce the protein. From protein, you have to take correct platform like recombinant platform you have to take. Then a product will come. The product you have to extract, and you have to purify the product, protein product. Then whether it is working similar to the human insulin or not, you have to cross check. If they are working, both are working. Normal insulin is working. If normal insulin is working and your recombinant insulin is also working in the same way, then you can go ahead for the patents or taking the approvals. So for nearly there are 35,000 genes are there. There are 3,500 protein products are there. There are thousands of enzymes are there. Hormones are there. They are not yet explored. Even in human being, it's not yet explored. Forget about other organisms. So much of work is there to deal with the things. So cell culture bioreactors are there, still they are in developing stage, and transgenic animals are there, still they are in developing stage. You can develop the transgenic fish, transgenic cattle, transgenic dogs, transgenic like that mammalian animals, and you can express the whatever the protein in them. So that xenotransplantation work is also is going ahead. Recently, mice is being developed with help of the diet protein that become transparent. So much of applications are there for collagen. So many of applications are there for no for joints. In between the joints, you have so much of collagen protein or chondrin material. Those materials also you can able to produce. So these are the methods how to produce these transgenic like the DNA microinjection methods, and retroviral mediated gene transfer methods, embryonic stem cell mediated gene transfer methods, and so several applications. I told that you can produce the interferons, you can produce the interleukins, cytokines, you can able to create the recombinant protein vaccines, you can go for tissue bone growth factors, clotting factors, monoclonal antibodies, diagnostic antibodies, therapeutic antibodies. So many things are there. So example, you can see these are the some of the products, some of the companies, some of the indications. So these are the list of the products. Like you can see the company name, it's a disease, alpha glucosidase. Company name is Gen. How many of you heard? How,
someone இது ஏதோ சொன்னா இது முன்னாடி வரும்ல அப்படியே சொல்றான்.
anyone wants to ask any question? Yeah, please mute yourself. See, these are the list of the recombinant proteins. You can see the product name on the left side, company, and what it is the application, what is the disease or something you can see. So alpha glucosidase, company name is Gen enzyme. It is involved in the Pompe's disease. So thousands of compounds are there.
Sir, excuse me, sir. Sir, you have unmuted your mic, sir. Please mute. Excuse me, sir. Share and unmute, sir.
Yeah. You have to give message. No, I have not seen. Oh, it's unmuted.
Yes, sir.
So
Yes, sir.
coming to this PowerPoint in the last page is important because rather than theory, the action is very important, because you are all young minds. you have to know the company's names, the products, and what is the disease they are treating. For example, a product name is thrombopoietin, company name is Pharmacia, and indication is thrombocytopenia. Next product, erythropoietin, company name is Amgen, and Amgen treatment is anemia. Third product,
Sir,
ancestrism,
excuse
Amgen
me, sir.
blood transportation.
Sir, excuse
Next
me,
is you
sir.
can
Sir,
see
your PPT is not shared.
PPT is not showing. One second.
Yes, sir. Dear participants, there is a technical issue. Please be still.
Audible? Is it audible?
Yes, sir. You are audible, sir.
Okay. See, what you have to get from this is, as a student, we listen so many lectures, but industrial point of lectures is very important because I worked in industry. I'm showing all these things to you. So here, these are the list of the recombinant origin. You can see these products, alpha glucosidase, company is Genzyme, and its indication, its application is Pompe's disease. So several components of companies are working on several products. They have patents, they have publications, they have the process, research and development process, and everything. And there is application for what purpose they are creating or no producing the products. Can have some kind of idea. So most of the therapeutic proteins, they are having very high commercial value from the biotechnology sector because these don't carry much side effects. For example, for the fourth product, you can see a company named Genvec. It is producing vascular endothelial growth factor for cardiovascular diseases. So the company has patent on that. The company has a very good patent on that. So it is going to keep on improvising the product from day one
Sir,
to day.
excuse me, sir.
Yeah.
Sir, excuse me, sir. Sir, your presentation is not visible, sir.
Not visible.
Yes, sir.
One second.
Yes, sir.
Now is it visible? My
Yes,
presentation?
sir. Yes, sir. It is
Yeah.
visible, sir.
It is some of the example you can see take here. For example, products alpha glucosidase. The company name is Genzyme. Its indication is its application is Pompe's disease. Like that, you can see few of the products here. So the product's name you have to, if you have a book, you can write one side for an example. So what is the product? What is how they have produced the product? What is the mechanism? What are the technical aspects in that? So how whether they have used the recombinant DNA technology or they have used the bioinformatics approach or they applied the biochemical approach or they applied the genetical approach or they carried the side-active mutagenesis or they used the codon usage tables. So how they have developed this product, which is very, very similar to natural one, which is produced inside our body. So the same protein which is there in our body, they are going to produce outside by using this biotechnological process. So the patent you have to understood the publication of the particular product, then patents, then how it is going to treat the disease, the molecular pathways, the biochemical pathways, the molecular pathways, and what are the side effects of the particular protein product that you have to make analysis. So several thousands of compounds are there. I cannot explain each and everything, but you have to make a rough sketch of the things. So if you want to apply for any of your company, for any of the positions over there, like scientist positions, or you can apply for technical officer positions or scientific officer positions, or simply you want to carry your JRF studies or HRF studies for to pursue for a PhD program, you should have some idea of the products. Then only you can able to apply for them as internal training persons. So internal trainship like that, so that you can have your vigorous growth job employment. At the same time, you can have a real knowledge what is the product and how it is going to applicable for all those things. So it is a heart of the thing where the lot of biotechnology business is going to happen. So first of all, as a student, you should have some kind of awareness. What is your purpose of learning the subject? You want to get only the academic excellence, then you can have do some MSc, then PhD, then you can go for academic side like universities or college side for teaching purpose. Or you want to establish your own company or industry, or you want to have your own entrepreneurship, then the teaching, simply teaching is not going to work. You have to get your own product. So want to get your own product, you have to combine your research skills, technical skills with this kind of stuff, and you have to start your own company by giving employment to thousands of people. And also you can give no your end product to common user, where you can have where you can have the patients can have access to clear their diseases. So these modern components, modern medicines, which are going to replace the allopathic medicines, which are having side effects, are going to be the day of the world. So now the biotechnology field is booming. The pharmacy sector is also booming. Life science are going like anything. So once you start your career in life science, once you crosses PhD work, or once you take the patent, or once you start your establishment of your own company, you need not to look back itself. So what is your ambition is very important. Rather go. So one idea is that it's very simple. Once you know the products line, what is the application? Second thing, in advanced countries, people are going to work day and night for these components. The same work you can carry in India. You can take the patents. You can start your own company, because most of the companies' patents expire by 25 years lifetime. So the patent which is expired in US or UK or Germany or Netherlands, wherever is there, if you find that very suitable product and which is going to take care of the Indian population, Indian communities, then you can start your own company. You can take the patent transfer from that particular company to your India. From India, you can start your own company and you can export to any other country. So that combination of the work is there. That particular point of view, you have to look to this recombinant therapeutic protein production. It helps for the patients. It helps for the health sector. It helps for the research and development. It helps for the academicians, for collaborations, and for the future growth of the each and every individual. So what you are going to express, that is very important. Without your involvement, without your own idea, you cannot do anything in this particular field. For example, myself in my PhD work, I have taken granulocyte colony stimulating factor, which is going to treat for neutropenia and breast cancer diseases. So one company came, Natco Pharma, and we collaborated with it, and we produced the product, and we published in several journals like Molecular Biotechnology, Process Engineering, for, Process Biochemistry. So many journals were published within 1 year. With real data. So that is went and circulated to international platform, and it is well versed. People have seen across the globe. They must very much shown interest. Most of the companies came forward to utilize our technology. So every time technology is going to shift from one phase to the other phase, and you have to track down which company is doing on the advanced technology. For example, mRNA vaccines are there right now. mRNA vaccines, you have to take care of things like in future, people are going to use CRISPR-Cas9 technology for gene editing. You have to take care of CRISPR-Cas9 technology. Some of you told that they want to use artificial intelligence. Take the help of the artificial intelligence and incorporate in these particular products, so that you can able to develop efficient products. Once efficient products are with you, it's there with you. You can able to go on any platform and you can challenge any of the scientific person or any of the community, and you can able to get grab your position, secured positions. So not only depending on theory, learning in the academics, writing the examinations. You have to try to interact with the real industrial persons. You have to seek, you have to understand their problems, how to overcome, how to challenge, how to overcome those problems, how to overcome those challenges. You have to think over, and you have to take the help of your teachers. You have to take the help of the mentors. You have to take help of the guides who are going to take care. And you have to meet the scientists who are working in the particular program across the globe. You have to visit their labs. You have to make internships. Then you can able to enrich your knowledge, and you can work on certain problems like these problems. And once you works out on that, that is going to be your experience. And with a real experienced person, nobody can replace a real experienced person. So that is the where no, a lot of industry is going to work out. So not only theory, participate in the conferences, workshops, learning the things and applying the things. You can generate your own idea, and you can generate your own product also, and you can approach the right candidate, right industry. So you can load your resume over there. If you are inexperienced person, they will train you. You're experienced person, they will offer you job. So it will work on like that. So I think I've given the most of the things. Now it is open for the session. You can ask questions so that I can answer. Okay, I am now stop sharing my PowerPoint.
Dear participants, now the session is open for the questions. If you have any questions, you can ask.
Please open your camera and try to ask with your open heart what you want to learn in the biotechnology sector. I am experienced in the cell biology, molecular biology, biochemistry, biotechnology. I'm also experienced programmer. I know the longest like COBOL, Fortran, Visual Basic, C++, Java, all these things I learned, Python, and all these things are part of my learning. Of course, anyone can learn. Even I taught for BTech biotechnology students higher engineering mathematics. So I know the differential equations and all those stuff. I learned I'm now planning to work in the artificial intelligence. So learning is a continuous process. While learning, you will get a lot of satisfaction also. So sky is the limit, because I came from Warangal to Hyderabad. From Hyderabad to I learned in ICRISAT, CCMB, CDFT, NIN, and several other institutions. Then I went to abroad as a scientist, biotechnology scientist. I've been paid the same amount of a American scientist in US, and a part of that I got double salary from Indian government. So at any time I can go. Still, I can go right now. After 10 years, 20 years also, I can go back to US. I can work with any pharmacy industry or biotechnology, or I can join in academicians as a professor, eminent professors. I can still give my services, because you cannot damage the knowledge. You can damage anything, but you cannot damage the knowledge of a person. So here is beautiful knowledge is there in terms of biotechnology, in terms of pharmacy, where you can develop the health products like recombinant therapeutic proteins. Lifelong you can learn, lifelong you can earn, lifelong you can earn honor. Don't think in terms of money, you can earn honor also. You can serve for many of the diseases, disorders, so that people will get benefit out of your contributions. As long as you are there, you will be honored. And after your perishment also, after 100 years also, people will refer your journals, they will refer your publications, they refer your experiments, they refer your patents, and still you will be honored. So there is no debt for that. Please, any one of you, please ask questions. So you are still very young stage. I know, I can understand. We are very young in learning the things, but learn with no. Always try to learn with the experts, so that no, in future your time will not be wasted. With experienced person, you can make small, less number of mistakes, and you can have a very bright career. And simple example I'm going to tell you. Just listen this example. In the year 2013, a young boy came to me to my lab. His name is Shubham Singh. He's from, I don't know actually, but he came to my lab. He's only BSc student, BSc second year student. At that time, I was the head of the department for 3 departments: biotechnology, microbiology, genetics, and zoology. 4 departments. I am the coordinator. At that time, we got the DBT Star College program for Nizam College. We got some 27 lakhs funding. We are developing the instrumentation, all those things. This boy from Uttar Pradesh, he had the word DBT Star College at Nizam College. He came. Then at that time, I got Ramlingasamy Fellow Project from US. A US green card hold citizenship, Senthil Rajagopal. He came to me and he worked under me with, I given him a fellowship of 1,50,000 rupees to him. It's a 85 lakhs rupees project. So in that project, they worked on the hepatic cancers, liver induced hepatic cancer, how alcohol is going to induce the liver cancers, what is the signaling mechanisms behind that they worked. And this work is published in the neurological sciences based journals. So 5 publications are given to this boy. And later he asked me to, sir, can you apply me to Harvard University? At BSc by asked me to apply for Harvard University. With a little bit no enthusiasm, with little bit encouragement, I applied to Harvard University, but they replied, you are in the waiting list. Then he applied for integrated PhD program at Indian Institute of Life Sciences, Pune. He selected for the program. There he met another mentor where he published 3 Nature articles. Then afterwards he approached me, sir, still I have some kind of interest to apply for Harvard University. Then applied for Harvard University. So he selected during the 2018 or 19 area to Harvard University. He joined as a postdoc study student and is continuing. So within the age of 25, 26 years, he became Harvard University scientist. Another girl in the 3 years back, she came approached. She's from MSA Environmental Science. I didn't know even about her. She worked for a few months with me, and she asked me, "Sir, I am going to Harvard University in neuroscience." So we applied together. She got 3 years back Harvard University postdoctoral position over there. So what you are planning is very important. Within 1 hour, we cannot give every idea. Right now, one person, Mohammed Siddiq, is working under me in neuroscience. He's working on IntelliCase system and no intelligent IntelliCase system and patch clamp system. This is a 1.5 crore rupees equipment is there in our lab. Our previous former professor Pratap Reddy, he brought with the DST funding. So we initialized, we opened the machine, we established the sensors, we spent some few thousands of rupees, and readily is working on the artificial intelligent case system. He's working on the sugar impact on the nervous system in the mouse. What is the effect of the sugar on the nervous system of the this mouse? So this mouse, how it is responding is going to be, we are going to calculate by using the artificial intelligence. Now is readily is now is going to move for Indian of Science for the nervous system and PhD. Then again he can go to the abroad like Harvard University, anywhere where neuroscience is like booming to understand the brain. Brain understanding, brain composition. The largest, biggest project is initiated by Obama on the brain project. It is a very highest project to understand how the brain is going to function inside the living organism. So you should have a concept. For the concept, you have to work. Then you can able to achieve anything. If you don't have concept, simply if you are just learning like a robot, you cannot get the idea. So that's what you have to interact with the eminent people, personalities. You have to learn, you have to grab their opportunities, then you have to make a move. It is not your way of thinking. You have to think in terms of the industry or academician, how they are thinking. Then you have to make a plan. Are you really understanding? Please raise your hands. I'm not seeing any one of you. I'm just feeling like I'm expressing to myself. What is your idea? Just debate. What is your idea? What you want to become or what you want to not become? You can have some kind of dialogue. No, you want to become a good biochemistry person. Yes, you can have a lot of options. You can learn HPLC, ORP, HPLC, all type of spectroscopic techniques. There is a lot of positions are in vacancy condition. Or you want to become a cloning person, you can learn molecular cloning techniques. There is huge demand is there for to produce the therapeutic proteins. You can apply for them in various industries in India or abroad. So many industries are working on the line. So many examples you can take. It's is a lifetime learning process. Don't think that I'm learning only for 3 months or 2 months like that, 2 years like that. It's a lifetime learning process. You have to express your ideas. Then only you can establish a bridge or connection. If you don't express, so people will not catch up you. Satish Babu Garu, any questions from the audience? I want to listen from them.
Yes,
One
sir.
or two questions, please.
Yes, sir.
Please
So
encourage
dear
them. Sir.
dear participants, so kindly ask the questions. If you have any questions, please ask, sir. Or if you want to clarify in your future goals,
Or if you want
what you
to
want to
come
do.
and work with us in Osmania University with IntelliCase system or patch clamp system or any of the advanced platform, like we have zebrafish model is there. We worked on the fluoride, and right now we are working on the ovarian cancers by using the artificial intelligence and CRISPR gene Cas mechanisms. If you want to participate, you can participate. It is zero cost.
So
Or
yes,
you want to
so
work,
sir,
you want to pursue in electron microscopy studies, nearby we have the electron microscopy facilities are there. It is also zero of cost. Anything you can plan. It comes with zero of cost, but at the same time, you can have a chance to become a world-known scientist. Right now, that Shubham Singh is earning more than 25-30 lakhs per month. And one more person, his name is Kiran. He's who was the first student of me in 2003. He is the chief CEO of Lonza company, a very big biotech company. He's the chief CEO of the Lonza company in London. One day after 25 years, he called me, sir, do you remember my name is Kiran? I told that you are my first student. What you are doing? So I am a CEO of the Lonza company in the London. I am looking after the operations of the entire Lonza company, which is a very big biotech company. So my monthly salary is much more better than anyone else. Sir, will you come and will you join with us? I told that I want to stay in India. It's nothing to deal with money, but you people, you can enjoy, you can grow up like anything. You want to give some kind of employment to any one of them, any students, please remember, call me. I will send my students to you like that. I told. So you should have some idea. Then only people will encourage and push you forward for your future. If you don't have idea, nobody will push you. If you want to eat chicken biryani, you have to say to your mama, I want chicken biryani. If you want to eat mutton biryani, you have to say to your mama, I want mutton biryani. If you want to eat prawn biryani, you have to select your option and to express to your mother. Then only the teacher or scientist is going to serve you. If you are just like that, nobody is going to serve anything. They don't have any curiosity to learn from you what you want. You have to express yourself. Who should have the akali? Akali means the no emptiness in the stomach. If you have hungry, then only you can ask. If you don't have hungry, you will be stable. Always a good research should have a lot of hungry for his food, like hungry for knowledge. Is it not?
yes, sir, it is absolutely. So any participants? So the sir is very cooperative, and if you want to plan for your future studies or
Sir, good afternoon,
doctoral
sir.
studies,
Sir, myself,
yes,
Dr. S. Purification, sir. Sir, your presentation is very good, sir. I think you are a multi-talented person. Sir, I had a question, sir. Sir, which one is best, the gene therapy or protein therapy, sir? Sir,
Both
your
are one
topic
and
is on protein therapy. What about gene therapy, sir? Can you give
both
few
are
suggestions, a few points
the
regarding gene therapy?
gene therapy is the upcoming therapies. Like doctors, if they want to cure the genetic disorders, they will go by gene therapy. But protein
Sir,
therapy
your
is like
voice is not
for
audible, sir.
diabetic patients. Protein therapy is the option. Like they are going to produce the proteins inside the laboratory, like hormones, enzymes, peptides, and they are going to treat the patients. It's one and same. Genetic therapy is the upcoming medicine. Maybe after 100 years, you can see gene therapy techniques to treat the diseases by using genes itself. Right now, you are treating the patient with the help of the medicines.
Sir,
Medicine is just
excuse
its
me, sir, your
organic
voice
product.
is not audible, sir.
Right now, our medicine, we are dealing with the patients. We are giving the allopathic medicines, which are the organic chemical compounds. These organic chemical compounds don't have any relation with our body, but still we are believing the medicine. we are taking the medicine. Of course, some of the compounds are very active compounds. They are curing the diseases. But in upcoming next generation medicine, the genetic therapies, the genes itself act as medicines. The right combination of the genes, they are going to clear the genetic diseases or disorders. Next is protein therapy. Your body itself is a combination of the all enzymes, hormones, cytokines, interleukins, interferons, like that. So need not to take extra medicine from outside like organic medicine. You have inside, you can take the protein molecules as therapeutic agents. They are going to cure 99.9% of the diseases without having any side effects. So these are the next 100 years. After 100 years, after 50 years, you are going to get these kind of technologies in the market. It is slowly replace the side effects based English medicines. Right now, we are using the English medicines. Slowly, it will get replaced by genetic therapies and therapeutic proteins. Because the companies has to come, no? So many companies, so many ideas, so many patents, so much of research work is still pending. That's what this field is, evergreen field. Patients, what they require a medicine without side effects, correct? If you promise, if you want to give the medicine without side effects, that will be it works. It is going to have commercial value. So genetic therapy is the line of the technology to deal with the genes, right? And proteins are the ultimate products which are helpful for treatment of the diseases or disorders. Can use both the things together.
Thank you, sir.
Yeah. Any other person? You want to pursue your career as protein engineer? Yes, it's possible. Or biochemical engineer is possible. Or biotechnology is possible. Molecular biologist is possible. Or you want to work with the doctors to cure the genetic disorders, it's possible. Any life science person can associate with themselves with the doctors, research organizations, and work on the genetic diseases or disorders. They will give the same salary as doctors. They will give same salary as scientists. If I am working in US, they will give the same salary as scientist of US. So
We will not ready.
Dear participants, so if you have any questions, you can ask sir. So he is willing to.
sir, my camera is not working. Still, I can able to listen your audio.
Yes, sir.
Yeah.
So dear participants, if you have any questions, you can ask. So sir is willing to answer your questions and queries. And if you want to share your future plans, research plans, or higher studies, so you can discuss with the sir. So he is a good source to discuss. The sir is ready to help you. So you can share your thoughts and knowledge, and you can clarify your future plans. So please interact with him.
Yeah, if they want to apply for JRF positions or SRF positions also, I will
Yes.
with the help of the systems biology or artificial intelligence or with the help of the bioinformatics tools, they can publish the data with Osmania University and they can go for any kind of higher studies.
Sir,
So yes, so
morning, sir.
Yeah,
Sir, am I audible?
yes,
Sir,
please tell your name and interact with the sir.
University, please.
so I'm Yuvashri from Satyabama College, sir. So being a bioinformatician, people are sharing me that India doesn't have scope for bioinformatics, sir. Is it true or not, sir?
It is completely false. When I was a student in 2003, I went to CCMB to ask the same question. They told the same thing, but we published the papers in scientific advances and put on usage. Okay. So then ultimately my paper was published in the Marker Biotechnology. Immediately, a professor from IIT Delhi, his name is Subhash Ghosh. He came on the all way to Hyderabad to meet me and to say congratulations that I worked on scientific advances and put on usage. That work still after 30 years also nobody has taken the work. That paper is there with me. If you want, I will send the paper. We developed 106 clones. Out of one clone is working very perfect. And Natco Pharma has taken the patent on that and granulocyte colony stimulating factor. See, people will say many things. Even after in 90s, people have told that after computers, you will not get jobs. But still, lakhs of jobs is generated in computers itself. So what you want, you have to get. That's it. No, don't listen to others. It's upcoming areas, all artificial intelligence or Python programming or Perl programming. So many things are there which you don't know. So if you ask any lay person how to reach the moon, he will not answer. But if you go to the NASA, they will send you to the NASA. They will send you to the moon. Okay, approach the right person so that you can have better dreams and will reach the dreams.
Thank you, sir. Thank you so much.
So if you want to communication or you want to clarify your doubts, please, I will give my email ID or you can collect from sir, Satyababu Rao, sir. You can collect from sir my mobile number or email ID and you can clarify the doubts. If you want to pursue some kind of training, you can come and approach to my lab. You can learn the techniques, whatever I told in the case system like that. You can apply for various positions for the neuroscience.
Of course, sir. Thank you, sir.
Yeah.
Dear participants, if you have any questions,
Sir,
you
good
can
morning.
ask the sir.
I am Dr. Jansi from Vellalar College, sir. Sir, is there any possibility for the students to take internship in your university?
Yeah, we'll take internships. Last time, 3 persons came from Vizag, Geetam University. 3 of them completed and they secured positions in the local biotech companies. And one girl, she got 174 rank in CSR. Right now, she's going to join under me itself to work on the CRISPR-Cas9 systems.
Okay, sir.
Yeah,
So
well, always internships are always friendly. They will not charge anything. If you want to learn the technology, anyone will offer. Like you can join with University of Hyderabad or BITS Pilani or Triple IT is there, or CCMB is there, CDFD is there, National Institute of Animal Biotechnology is there. So many other institutions like Bharat Biotech is there, Shanta Biotech is there. So many are there.
Okay, sir.
Yeah, but the thing is you have to learn. The only thing is that I have to learn. The concept has to be there within oneself, and you can apply for various postdoc positions also abroad. So many are there for real technology, for the real persons. There are so many vacancy positions are there available.
Okay, sir. So can you share your mail ID, please?
Sure, it is Venbio Venkatesh Bio. Bio means biology. venbio@gmail.com. Venbio. VENBIO.
Oh, okay, sir.
Yeah.
Okay, thank you, sir.
Yeah. Well.
So any other questions from the participants? Or any doubts you want to clarify? It's okay. So if you do not have any questions, we can move forward. So thank you, sir. So thank you so much for your efforts. So now it's a time to deliver a vote of thanks. So now I would like to call upon Dr. S. Vadivokrasi, Assistant Professor, Department of Biochemistry, to propose vote of thanks. Welcome you, ma'am.
Good afternoon to all. As we wrap up day 3 of our special online lecture series, I am both honored and delighted to address you and extend our deepest gratitude. Today has been truly inspiring and impactful. Thanks to the collective effort and unwavering support of many dedicated individuals and teams. First and foremost, our heartfelt thanks goes to late Lion Dr. K.S.Rangasamy MJF, the esteemed founder, and Mr. R. Srinivasan, chairman of Education Institutions. Your steadfast guidance and support have been pivotal in bringing this event to life. We also extend our sincere appreciation to Mr. Sachin Srinivasan, Vice Chairman of KSR Educational Institutions, and Mrs. Kavita Srinivasan, Executive Director of KSRCAS, for their invaluable contributions. Our gratitude also extends to Dr. V. Padmanabhan, Principal of KSRCAS, whose encouragement and support have fostered an environment conducive to this event's success. We are thankful to Dr. M. Prasanna Rajesh Kumar, Vice Principal, and Dr. G. Saravanan, Dean of School of Sciences and Humanities, for their essential role in organizing and supporting this series. A special thanks goes to Dr. J. Venkateshwara Rao, Professor in Zoology, Osmania University, Hyderabad, for your enlightening presentation today. Your deep knowledge and passion for the subject were truly inspiring, and we are grateful for the clarity you brought to our discussions. Special thanks, sir. We also want to express our appreciation to Dr. Suresh, the librarian at KSRCAS, for facilitating our online meetings through Microsoft Teams. Your efforts have ensured the smooth conduct of this event. Last but certainly not least, we want to thank all our attendees, participants, and contributors. Your engagement and active participation are key to making today's session meaningful and impactful. As we eagerly anticipate J4, gratitude to everyone involved. Your dedication and support are what make this series a success. And we look forward to seeing you all tomorrow for day 4 of our lecture series. Thank you all.
Thank you, ma'am. Thank you for your kind words. Thank you so much. So thank you all the participants. So dear participants, the attendance link will be posted in group. So kindly fulfill that feedback form and attendance form. Thank you. Thank you very much. And we look forward to meet you on tomorrow, day 4. Thank you. So thank you, sir. Thank you so much, sir.
Thank you, Satyababu Garu. Thank you
Thank
very
you,
much
sir.
for the patient listens of your students. I wish them all the best for their future endeavors. The young scientists, the young minds has to grow up and has to become the future leaders.
Thank you, sir. Thank you so much, sir.
Thank you, sir.
Thank you, sir. Thank you very much.
So dear participant, the link will be posted in the group. So you can fill that form. Thank you.